In the last class, we discussed about the necessity of light for photosynthesis and the necessity of carbon dioxide for photosynthesis. Yes, John Ingenhousz proposed that the light is necessary for photosynthesis, and along with that, he proposed another statement, that is, the photosynthesis can be carried out by only green parts of the plant. If this is true, we have some doubts. Yes, if we have some questions. Yes, with this statement, with this proposal, there arise some of the questions. Yes, what are those questions? Here, the newly formed leaves. The newly formed leaves in a plant, they are in dark red color. Don't they perform photosynthesis? Yes. Next, another question. Yes, so some of the ornamental plants contain some of the leaves with reddish color and other colors, and don't they perform photosynthesis? Yes. And next, if the green color is responsible for photosynthesis, then why some of the green colored animals and birds don't perform photosynthesis? Yes, these are all the questions we have. But the green colored, all the green colored animals and birds, they do not perform photosynthesis. And in the plants which contain the red colored leaves also perform photosynthesis. Yes, that means, so there is a special thing present inside the plants, yes, which is responsible for photosynthesis. Yes, that is responsible for green color as well as that is responsible for photosynthesis. Yes, so that was not known at the time of Ingenhousz. Right. So next, after 4 decades after the proposition of Ingenhousz statement, yes, there arrived 2 scientists. Those are Pelletier and Caventou. Yes, in 1817. So Ingenhousz proposed in 1779. Yes, so these Pelletier and Caventou, after 4 decades, yes, 1817, they observed the green colored substance. They extracted the green colored substance from the leaves. So, and they named that green colored substance as chlorophyll. Yes, chlorophyll means green leaf. Chlorophyll, the word chlorophyll means green leaf, right? So here chlorophyll is responsible for photosynthesis, they said. Yes, the Pelletier and Caventou said that chlorophyll is responsible for photosynthesis, right? So here the substance which is necessary for photosynthesis is not only the green color, the substance is chlorophyll. Yes, and along with chlorophyll, inside the leaf there are other pigments which are accessory pigments, accessory photosynthetic pigments. Chlorophyll is a photosynthetic pigment. Along with that, there are other photosynthetic pigments are also present, which are called accessory photosynthetic pigments. Those are carotenoids, phycobilins, and anthocyanins are also there. Yes, which are helpful in photosynthesis. Yes. So next, and where that chlorophyll is present? Yes, according to Pelletier and Caventou, chlorophyll is the substance which is necessary for green color of the leaves, and that is responsible for photosynthesis, and where it is present. And he believed that the chlorophyll is spreaded throughout the leaf. The chlorophyll is spreaded throughout the leaf. But after so many years, after 6 decades, in 1883, Julius von Sachs, there is another scientist, Julius von Sachs, he proposed that, and he observed the leaf structure, and he proposed, and he said that yes, the chlorophyll is not spreaded all over the leaf. It is present in a specialized cell organelle, not in a cell organelle called chloroplast. Inside the plant cells, inside the cells of the leaf, so many cell organelles are present. Among all the cell organelles, yes, one specialized cell organelle, which is nothing but chloroplast. So that contains chlorophyll, he said. Yes, so to know where the chloroplast is present and how the structure of the leaf, yes, to know that we have to observe the T.S. of leaf. T.S. of leaf, that is transverse section of leaf. So let us see this. Yes, so this, if you take this leaf, yes, it is the upper surface. This is the upper surface of the leaf, and this is the lower surface of the leaf, right? So the upper surface is this one, and lower surface is this one. Yes, so in middle between upper surface and lower surface, the middle, this part is present, right? Yes, the smaller part, just very thinner part, yes, this is zoomed and this is this one. Okay, so this is the upper surface is this one and lower surface is this one. Yes, in middle what are present, let us see, right? So here in TS of leaf, let us observe, here it is upper surface and it is lower surface. This is called upper epidermis. Epidermis, dermis means just like covering. Yes, epi means outer surface, outer side. Yes, so here this is upper epidermis and this is lower epidermis, and this upper epidermis is covered by a tough layer called cuticle. A tough layer called cuticle, and the lower epidermis also covered by a tough layer called cuticle. Yes, as we already known that in leaf stomata are present, and where the stomata are present, at the lower surface of the leaf stomata are present. Yes, so here this is the lower epidermis, and here in this lower epidermis stomata are present. Yes, so here these are the stomatal openings, and these stomatal openings are guarded by specialized cells called guard cells. These are the guard cells and it is stomatal opening. Yes, so here also one more stomata we can draw. Yes, here also it is a stomatal opening and these are the guard cells. Stomatal opening and closing is controlled by guard cells which are present in the lower epidermis of the leaf. Yes, so upper epidermis and lower epidermis are covered by cuticle, and at the lower epidermis stomata are present and those are guarded by guard cells. And between these two surfaces, between upper epidermis and the lower epidermis, there is a mesophyll cells are present. The mesophyll cells are present. This is totally called mesophyll. Yes, so mesophyll cells. Yes, in the mesophyll cell, the specialized structure is present, which are called the vascular bundles. Vascular bundles. And we know what are vascular bundles. So vascular bundles are nothing but xylem and phloem. So here if you observe, yes, this part at the lower, from the lower surface we can observe these veins and the main midrib. Yes, we can observe the midrib. Yes, so this midrib is like this. So here this is the vascular bundle, this part, this smaller part, yes, this midrib part we are drawing here. This is the one, right? So these are the vascular bundles. So inside the vascular bundles what are present? Xylem and phloem are present. At the upper surface of this vascular bundle, this is xylem, this is xylem, and at the lower surface, this is phloem. And we know that xylem is responsible for the transport of water and minerals to every part of the body, every part of the plant body, and phloem is responsible for the transport of food materials from leaves to every part of the plant body, right? As we know that. And here in this there are the mesophyll cells. Yes, there are two types of mesophyll cells. Those are the mesophyll cells we can also call as parenchyma. Two types of parenchyma are present. Yes, one is palisade parenchyma and another is spongy parenchyma. So here at the top, at the upper surface of the leaf, the palisade parenchyma is present. That is of 1 or 2 rows of palisade parenchyma is present, and here the cells are closely arranged. The cells are closely packed. Palisade parenchyma is present, and here at the lower surface of the leaf, yes, the parenchyma is loosely arranged, which we call spongy parenchyma, which we call spongy parenchyma. The upper surface contains palisade parenchyma. Here all the cells are closely packed, and here at the bottom surface or at the lower surface, all these cells are loosely arranged, and this is called a spongy parenchyma. Okay, so these are all the cells of the leaf. These are all the cells of the leaf, and each cell contains at least 40 to 100. Each cell contains 40 to 100 chloroplasts in it. Yes, each cell contains 40 to 100 chloroplasts in it. So these all dots we can call as chloroplast. So the upper surface contains palisade parenchyma. Yes, and here at the lower surface, the parenchymal cells are loosely arranged, and that is called spongy parenchyma. In each mesophyll cell, yes, 40 to 100 chloroplasts are present, and inside this chloroplast, actual photosynthesis will be taken place. So these are all the chloroplasts. In these chloroplasts, actual photosynthesis will be taking place, and the number of chloroplasts is more in palisade parenchyma when compared to spongy parenchyma. Yes, and here the spongy parenchyma is arranged loosely, and what is the reason for that? Here the air can be entered. The air can be entered through stomata, right? Yes, carbon dioxide can be entered through stomata into the leaf, and which is necessary for photosynthesis. Yes, so for the entry of carbon dioxide easily, the spongy parenchyma is arranged loosely. So that is the reason spongy parenchyma is loosely arranged. Spongy parenchyma contains loosely arranged cells. It is upper epidermis, it is lower epidermis. Yes, upper epidermis and lower epidermis are covered by cuticle layer. Yes, and inside this a specialized structure is called the vascular bundles, and in between these two, upper epidermis and lower epidermis, parenchymal cells or mesophyll cells are present. Though these are of two types, those are palisade parenchyma and spongy parenchyma, and each parenchymal cell contains 40 to 100 chloroplasts, and there the actual photosynthesis will be taken place. Okay, if we want to observe the process of photosynthesis, so we have to extract this chloroplast. Yes, so here first Pelletier and Caventou, what they did, they extracted the green substance from the leaf. So that is very easy thing. That's very easy thing, just to extract the green substance. What is that green substance? They have extracted it. Yes, and here next another person, Julius von Sachs, he said that chlorophyll is not spreaded over the leaf, and it is present in a specialized cell organelle called a chloroplast. So extraction of chloroplast is not easy as extracting the chlorophyll. Chlorophyll can be extracted easily, but extraction of chloroplast is very difficult task, right? So if you tear a leaf, along with this leaf, yes, the chloroplast also will be broken. The chloroplasts are also will be broken into pieces, because in each parenchymal cells 40 to 100 chloroplasts are present. 40 to 100 chloroplasts are present, and everywhere the chloroplasts are present inside the leaf. So if you break it, if you tear the leaf, chloroplasts also will be broken. So if you want to observe the photosynthesis, the process of photosynthesis clearly, you have to get the chloroplast perfectly without breaking or without damage. We have to extract the chloroplast perfectly. So that was done for the first time in 1954 by a scientist called Daniel I. Arnon. Daniel I. Arnon was the scientist. He extracted the chloroplast for the first time without any damage. Yes, so let us observe the structure of a chloroplast. This chloroplast can be seen not only in plants and in all the photosynthetic organisms like chlorella, chlamydomonas, yes, spirogyra. So like this, in all these organisms, the chloroplasts can be seen. Yes, so these chloroplasts are not in same shape in all these organisms. Yes, so here they have different shapes. If you take a chlorella, in chlorella the chloroplasts are plate shaped. Yes, in the chlamydomonas the chloroplasts are cup shaped. Yes, in Spirogyra the chloroplasts are ribbon shaped. Yes, if you take, we are talking about the plants here. So in the higher plants the chloroplasts are in oval shaped or ovoidal shaped or spherical shaped. Yes, so let us observe the structure of chloroplast of plant cell. Right, so here we have already seen that in these mesophyll cells, in each cell the chloroplasts are present. So this one chloroplast, so this one we are taking here. Yes, so this one chloroplast we are taking, and what is the structure of chloroplast that we are going to observe here. Yes, so chloroplast is in ovoidal or circular shape, and its size is 4 to 5 micrometers. So its size is 4 to 5 micrometers. Yes, so it is a membranous structure. Yes, chloroplast is a membranous structure. It contains three membranes. Yes, this is the outer membrane. Yes, outer membrane. This outer membrane is completely permeable membrane, so it allows all the materials to flow inside. Yes, so it is outer membrane, is permeable membrane, and the next second membrane is selectively permeable membrane. Second membrane is selectively permeable membrane. And next, the third membrane of the chloroplast is modified into the stalk-like structures. Right? So here let us see the third membrane of the chloroplast is modified into stalk-like structures. Yes, so this is the stalk. Stalk means a pile of, just if you take while you are playing caroms. Yes, so you arrange the coins like a pile or like a stack, one on the other. In the same manner here also, so this is the sack like structure. It is a sack. So these are all sacks. Yes, and all these sacks are arranged together and it made into a stack like structure. Yes, so it is a stack like structure with several sack like structures. Right, so here there are several stalk like structures present in the chloroplast. Yes, and these stalk like structures are connected with another one. Yes, so here these stalk like structures are called as grana. Stalk-like structures are called as grana. That means the third layer or the third membrane is modified as grana. Right? Next, here, so there is a fluid-filled place is present. Inside this, the matrix is present. Other than these three layers, other than these three membranes, there is a fluid-filled place is present inside the chloroplast. That is called stroma. And here actual enzymatic reactions take place, and here only the starch will be produced. And it contains, stroma contains DNA also. Yes, the chloroplast is a semi-autonomous cell organelle. So it contains even DNA and ribosomes also. Yes, so that is why all the enzymatic reactions will be taken place in this stroma. What are the parts of chloroplast now? So it contains three layers. The outer layer is permeable membrane. Yes, and the second layer is selectively permeable membrane, and the third membrane is converted into stalk-like structures which are called as grana. Yes, and let us extract this one stalk. Yes, if you take this, yes, it is made up of so many sac-like structures. Yes, this is one granum. Yes, singular, it is one granum, right? So this is one granum. So like this, several granum are present. Then the plural is grana, singular is granum. Yes. So each granum is made up of 20 to 60 thylakoids. 20 to 60 thylakoids. Yes, each granum contains 20 to 60 thylakoids. So what are thylakoids here? So this is one thylakoid and it is another thylakoid. Yes, just as I said just now in caroms, yes, one coin is one thylakoid, another coin is another thylakoid. Just like that, on one coin, another coin, just we are placing a coin on the other. In the same manner, each coin represents a thylakoid here, one thylakoid here. So all these thylakoids are collectively forming granum. Yes, mostly 20 to 60 thylakoids are arranged together and they form one granum. And all these granums are arranged like this. So they form grana. They form grana, right? So in this thylakoid, so what are present inside in this thylakoid? If you extract one thylakoid here, if you extract one thylakoid, yes, it contains one thylakoid lumen. Yes, so in this there are some granule-like structures are present. In this thylakoid, some granule-like structures are present. Yes, these granule-like structures are called as quantasomes. These granule-like structures are called as quantasomes. Yes, and what are present in these quantasomes? Yes, so in these quantasomes, the actual chlorophyll molecules are present. Yes, in these quantasomes, the actual chlorophyll molecules are present. Chlorophyll molecules and carotenoid molecules are present. So here in chlorophyll, the chlorophyll in the plant cells is of two types, that is chlorophyll A and chlorophyll B. Chlorophyll A is blue-green in color and chlorophyll B is yellow-green in color. Yes, so here in these quantasomes, chlorophyll A and chlorophyll B are present. Along with these chlorophyll molecules, carotenoid molecules are also present in these quantasomes. That means here almost 250 to 400 molecules of 250 to 400 molecules of pigments. Yes, that means the chlorophyll A, chlorophyll B and carotenoids. So like that, pigment molecules are of almost 250 to 400 clubbed together and they form one quantasome. Yes, in each quantasome, how many molecules are present? 250 to 400 molecules are present, and what are they? Chlorophyll A, chlorophyll B and carotenoids. Yes, and these quantasomes are also called as photosynthetic unit, and those are also called as light harvesting complex. Yes, so each quantasome is also called as a photosynthetic unit and light harvesting complex. That means where the chlorophyll is present now, inside the chloroplast granum is present, and inside the granum thylakoids are present, and in the lumen of the thylakoid, quantasomes are present, and those quantasomes contain chlorophyll molecules. Yes, these chlorophyll molecules are responsible for harvesting the light. Harvesting the light means trapping the light energy. Yes, so in this manner the light energy will be trapped. So the light energy will be trapped in granum. Yes, the light energy will be trapped in granum. And why the light energy will be trapped here? So in this only the chlorophyll molecules are present. The light harvesting complex is present. Yes, that means the quantasomes are present. So that is why they trap the sunlight and they give. That means they transfer the light energy. Right? So here that light energy will be trapped, and these chlorophylls will be activated, and then that light energy will be transferred to stroma. And that light energy will be transferred to stroma. And here other enzymatic reactions will be taken place. So here if you observe the chlorophyll molecule, and it has the close resemblance with the hemoglobin of our body, of our blood. What is the main function of hemoglobin? It transports oxygen to every part of the body, right? So hemoglobin is the oxygen carrier which gives us energy. Yes, in the same manner, if you take plants, in plants chlorophyll is present. Chlorophyll also traps the sunlight, which is nothing but energy, and it traps the sunlight and it gives to the stroma, right? So here the main difference between the chlorophyll and hemoglobin is hemoglobin contains iron as a principal element, and this chlorophyll contains magnesium as a principal element. So this is about the structure of a chloroplast. During photosynthesis, so there are several events carried out in chloroplast. So those events are: first event, conversion of light energy to chemical energy; next, splitting of water molecule; and next, reduction of carbon dioxide to carbohydrates. Yes, these are the three events. These are the three main events take place in photosynthesis. Those are conversion of light energy. So the light energy will be trapped by the photosynthetic unit or light harvesting complex, nothing but quantazome. Yes, so those absorb the light energy and they convert that light energy into chemical energy. And next, with that energy, what happens here? Water molecule will be splitted into their constituent ions. That means splitting of water molecule will be taken place. And next, reduction of carbon dioxide to carbohydrates. So that means carbon dioxide will be reduced to carbohydrates. Yes, these are the three main events that are taking place in chloroplast. So here all these events must be initiated by some energy, right? So every event should, or every reaction must include some energy. So that energy will be given by light, right? So for this, for some of the events, the light is necessary. The reactions of the chloroplast which require light energy, those are called as light reactions. Those are called as light reactions or light dependent reactions. Yes, so which part of the chloroplast which part of the chloroplast absorb light energy? Grana. Yes, grana contains thylakoids, and inside the thylakoids the chlorophyll molecules are present. Chlorophyll molecules and all the pigment molecules are present, and they act as a light harvesting complex. Yes, so they get the light energy. That means all the light dependent reactions will be carried out in grana of the chloroplast. So clear? Right. Next, and some of the reactions do not require light energy. That means those type of reactions, the reactions which do not require light energy, are called as light independent reactions or dark reactions. Yes, so those dark reactions will be taken place, or light independent reactions taken place in stroma of the chloroplast. So let us see, in light, the light reactions take place in grana. So in the grana of chloroplast, light reactions take place, and dark reactions take place in stroma of chloroplast, right? So let us see the mechanism of photosynthesis. There are two reactions in photosynthesis. Yes, one is light reaction, another is dark reaction. So let us see first light reaction. Yes, so light reaction or light dependent reaction. So why it is called light dependent reaction? So the series of reactions will be carried out in this, but so these reactions are started, or they are initiated by the light energy. They are initiated by the light energy. That is why this is called as a light dependent reaction. Yes, so here in this there are three steps. Yes, in light reaction we can write the light reaction in three steps. The first, step 1, activation of chlorophyll. Step 1 is activation of chlorophyll. Okay, what is activation of chlorophyll? So already we have discussed that. Yes, so it is a thylakoid. So in this thylakoid, in the lumen of the thylakoid, the light harvesting complex is present. The light harvesting complex is present, or quantasomes are present, and inside this several, that means 250 to 400 pigment molecules are grouped together and they form into light harvesting complex, and which we are calling as quantasome or light harvesting complex or photosynthetic unit. Yes, so here the light energy, when the sunlight falls on the leaves, what will be happen? These light harvesting complexes, they absorb the sunlight. They absorb the sunlight and they by the absorption of that sunlight, what will be happen here? The chlorophyll is present, and that chlorophyll will be activated. Chlorophyll will be activated by absorbing the sunlight. So that is the first step, activation of chlorophyll. Yes, next, and the second step, step 2, in this what happens? It is splitting of water molecule. Splitting of water molecule. Yes, so what is the second step? Splitting of water molecule. Just here in the first step, chlorophyll is activated. That means it has the light energy. It has the light energy in it. Yes, it contains energy in it, and with that energy, what happens here? Water, H2O, yes, that will be splitted into its constituent ions like H plus and OH minus. H plus and OH minus. Yes, so splitting of water molecule has been taken place in this second step. Yes, that is in presence of sunlight this will be happened. Yes, due to the energy trapped by the chlorophyll, and due to that energy this splitting of water molecule will be taken place, right? And next, step 3, formation of assimilatory powers. Formation of assimilatory powers. Yes, the third step is formation of assimilatory powers. And here in this, what happened here? H2O, water is splitted into its constituent ions like H plus and OH minus. So the next, these H plus and OH minus ions will be entered into the next step. Yes, so here OH minus ions are converted into oxygen and water again. Oxygen and water again. Yes, so this water can be utilized by the plant for its uses, for its needs, and this oxygen will be released outside. So these H plus and OH minus from step 2 are entered into step 3. From these two ions, OH minus ion, through the series of reactions, it has formed oxygen and water. So this water is utilized by the plant itself, and this oxygen is released outside. Then what about H plus ion? In the leaf itself, NADP is present. NADP is present. Yes, so this H plus is going to add with this NADP. Yes, what is that? Nicotinamide adenine dinucleotide phosphate. Yes, nicotinamide adenine dinucleotide phosphate. So with this H plus is going to be added. So when this is added to NADP, yes, then what happened here? NADPH is formed. NADPH is formed. Then what is this? Yes, reduced nicotinamide adenine dinucleotide phosphate. Yes, as already we know that, yes, when hydrogen is added, then that is called reduction. Yes, so this is nicotinamide adenine dinucleotide phosphate. So when it is added with the hydrogen ion, then it will be converted into NADPH. Yes, it is an assimilatory power. That means it is ready to use. Assimilatory power is nothing but ready to use. And next, it is another reaction. And next, another reaction will be taken place. That is ADP, adenosine diphosphate. Adenosine diphosphate is added with one inorganic phosphate. PI or IP also we can write. Yes, so one phosphate, inorganic phosphate. By the addition of inorganic phosphate to ADP, then it will be converted into ATP. It will be converted into ATP. So here ADP, adenosine diphosphate. So two phosphates are present, and for this one inorganic phosphate is added. Then it will be converted into ATP, that is adenosine triphosphate. So three phosphate molecules are present in this. Right. So here NADPH and ATP are the assimilatory powers. So these assimilatory powers are formed at the end of the light reaction. So let us see one more time. So the light dependent reaction, yes, in this light dependent reaction, the reaction is initiated by the light energy. So that is why it is called a light dependent reaction or light reaction. So this reaction is going to take place in grana of thylakoid. Yes sir, the grana of chloroplast. This reaction is going to happen in grana of chloroplast. Yes, here it has three steps. Light dependent reaction has three steps. One is activation of chlorophyll. By the absorption of light energy, chlorophyll will be activated. And next step 2, in the second step, splitting of water molecule into its constituent ions. By the energy absorbed from the sunlight, yes, this water molecule will be split into its constituent ions like H plus and OH minus. Then these two ions will enter into the third step, that is formation of assimilatory powers. Yes, so in this hydroxyl ion undergoes several reactions to form oxygen and water. Yes, this water will be utilized by the plant itself and oxygen will be released outside. Then here H plus OH minus is used here, and H plus ion, this H plus ion is added with NADP and it forms reduced NADP, that means NADPH. Next ADP is added with one inorganic phosphate and it forms ATP. So at the end of the light reaction, what are formed? NADPH and ATP are formed. These two are assimilatory powers. The next light independent reaction or dark reaction or biosynthetic phase. So this is called a photochemical phase and it is called a biosynthetic phase. Yes, so here the synthesis of starch molecules will be taken place and glucose molecules will be taken place in dark reaction. So here it doesn't mean that dark reaction doesn't mean that it will be happened or it takes place only in dark conditions. Yes, it doesn't need light, that's it. Yes, in absence of light the reactions are taking place in this. Yes, so this dark reaction is also called as Kelvin cycle, and here this reaction is also called as Hill's reaction. Yes, the splitting of water molecules. Yes, this was proposed by Robert Hill. So that is why this is called as Hill's reaction. And here this light independent reaction is also called as a Kelvin cycle. Yes, so this is an anabolic process. It is an anabolic process that we have two types of processes, anabolic process and catabolic process. Yes, so it is an anabolic process in which the smaller molecules will be converted into larger molecules. Yes, the simpler molecules will be converted into complex molecules. That is what happening in photosynthesis. The simpler molecules like carbon dioxide, water and minerals will be converted into the complex molecules like starch, right? So here also carbon and carbon material from carbon dioxide. So carbon enters into this phase in the form of carbon dioxide, right? So that carbon is converting into carbohydrates or glucose or starch. Yes, from the simpler molecules, the complex molecules are forming here. So this is called anabolic process. Catabolic process means digestion. So that we will learn in heterotrophic nutrition, right? So here it is an anabolic process. In this, the simpler molecules are converting into complex molecules like carbon is converting into glucose from carbon dioxide to glucose. Yes, so here the energy used is the assimilatory powers. So which are formed at the end of the light reaction, those are assimilatory powers, NADPH and ATP. So those were used here. That means ATP and NADPH are used as energy sources to convert carbon dioxide to glucose. So here what is happening here? Reduction of carbon dioxide will be taken place. So what is the first event? Light energy trapping, conversion of light energy into chemical energy, and next splitting of water molecule. Those two events have been taken place in light reaction. And the next reaction, what is that? Yes, so that is reduction of carbon dioxide to carbohydrates. So that is taking place in dark reaction or in light independent reaction, and this total reaction takes place in stroma of the chloroplast. Yes, so where the complete food materials will be prepared. Yes, an enzyme is very helpful in this reaction, which is RUBP, ribulose 1,5-bisphosphate, ribulose 1,5-bisphosphate. Yes, this is the enzyme which is used in this reaction, in this dark reaction. So at the end of the dark reaction, what will be formed? Starch will be formed. So the end product of dark reaction is starch, and the end products of light reaction are NADPH and ATP, which are assimilatory powers. Yes, dark reaction is not a simple reaction here. So it undergoes numerous reactions and numerous steps it has. Yes, it contains a number of reactions, and which will be learned by you in your higher studies in Kelvin cycle. Yes, so this dark reaction is also called as a Kelvin cycle, right? So this is about the photosynthesis, the mechanism of photosynthesis, and how the autotrophs perform their photosynthesis and they prepare their food materials. Yes, and in the next class we will learn about the heterotrophic nutrition.